everyone. Myself Manju Sharma from Suresh Canviat University, Jagatpura, Jaipur. The department is School of Agriculture. The subject is diseases of field and horticultural crops and their management. So today we are going to discuss about rice diseases. Here we are talking about major diseases of rice and their management. Rice diseases. Here we will talk about blast disease, bacterial blight disease, thyroid disease, and brown spot disease. Blast disease that is caused by Pyricularia gracia. So first is blast of paddy. Blast symptom can occur on leaves, leaf collars, knots and panicles. Leaf blast. The lesions are elliptical or spindle shaped with brown border and gray centers. Collar blast or knot blast occurs when the pathogen infects the collar that can ultimately kill the entire leaf blade. Then, the neck blast occurs when the pathogen infects the neck of the panicle. The infected neck is griddled by a grayish-brown lesions and the panicle falls over if the infection is severe. If neck blast occurs before the milk stage, so the entire panicle may die prematurely, leaving it white and completely unfilled. The fungus produce two toxins like mu picolinic acid and pyricularin. Like these are the spores or pathogen which cause the blast disease. Factors favorable for blast like upland condition, extended dew periods, high relative humidity, intermittent rain shower or drizzle, and high nitrogen fertilizer. So the spores are released by dew or rain and are carried in the air to other plants. Then the fungus produce more spores. Then the airborne spores called Conidia land on rice leaves. Then the spores germinate and the fungus penetrate the leaf surface or enter the leaf through the stomata. After that, the fungus grows and produce leaf spores after 4 to 5 days. Then again, the spores are released by dew or rain and are carried in the air to other plants. And then the fungus produces more spores. Then these more spores they will be airborne and their spread will be through the air. So the airborne spores that will be called Conidia land on the rice leaves. After that, the spore will be again germinate and the fungus penetrates the leaf surface or enters the leaf through the stomata. So, their management like use of resistant cultivars, efficient use of nitrogen fertilizers, soil amendments with silica, and spray beam tricyclazole, melanin biosynthesis inhibitor fungicide for effective control. So basically, the blast disease, which will be like different because the which part of the plant is infected by the pathogen. So the name given according to the infected part. Like if the collar region is infected, then it will be known as collar blast. Then uh, if the leaf is infected or the symptoms which is showing on the leaf, then it will be called leaf blast. If node are infected or symptoms which are showing on the leaf, sorry node, then it will be called the node blast. And if the spots are on the neck, then it will be called neck blast. Then the next disease is the bacterial blight disease, which goes by Genthomonas oryzae or PV oryza. So there is the crease occurs in early stage seedling wilt plants, whiters and dies up. In later stage, blighting starts from the tip of leaves to the base. The straw turn yellow to white, then partially field grains and appearance of bacterial oze, a milky or opaque dewdrop on young lesions. So these are the symptoms, basically the part of the leaves or the upper part which will be whiter in color. So how we can manage the disease? The use of resistant varieties, seed treatment with bleaching powder, that is 100 microgram per ml, and zinc sulfate, it should be 2%. Field sanitation, such as removing weed host, rice straw, ratoons, and volunteer seedlings. Use balanced fertilizer doses, especially nitrogen. Proper plant spacing should be there. Then spray of Bleetox, copper oxychloride, 50 vegetable alternate with streptocycline with the appearance of disease. Then the next disease is the brown spot disease, which goes by Helminthosporium oryza, then Bipolaris oryzae, then Cochlebolus meabenus. The disease was responsible for Great Bengal famine in 1943. The total deaths current estimate that is 2.1 million. Central Rice Research Institute CRRI 23rd April 1946 Vidyadharpur Odisha. These are the symptoms. The fungus attacks the crop from seedling to milky stage in main field. Symptoms appear as minute spot on the coleoptile, leaf blade, leaf sheath, and bloom, being most prominent on the leaf blade and glumes. The spores become cylindrical or oval, dark brown with yellow halo later, becoming circular. Then the several spores coalesce and the leaf dries up. So the seedlings die and affected nurseries can be often recognized from a distance by stressed appearance. Dark brown or black spots also appear on blooms, leading to grain discoloration. It causes failure of seed germination, seedling mortality, and reduce the grain quality and weight. So these are the symptoms of the disease. Like on the leaf, there will be a small the spores which will be becoming large in size, and this time the whole leaf which will be entirely infected, and then it will be fall down. Then the favorable conditions like temperature of 25 to 30 degrees centigrade with relative humidity above 80% are highly favorable. Then excess of nitrogen aggravates the disease severely. Then the disease cycle. The disease is transmitted by the infected seeds. After that, the disease spores germinate and enter the seedling roots or polyptile. Then, as the rice grows, spores are formed on leaf spore. These spores are then blown to the leaves and panicles of the other plants. After that, the spores germinate and infect the plant leaves of panicles. Then grain becomes infected. The grain will be become infected when the disease develop in the panicles. So after that, and after the development of the disease in the panicles, basically it will be again transmitted by rice infected seed, then rise to the disease spores which will be germinate and enter the seedling roots or coleoptile. Infected seeds and staples are the most common source of primary infection. The conidia present The conidia present on infected grain and mycelium in the infected tissue are viable for 2 to 3 years, and the airborne conidia infect the plants both in nursery and so the conidia will be present on infected grain and mycelium in the infected tissue are viable for 2 to 3 years. Airborne conidia infect the plant both in nursery and in main field. Infected seeds and staples are the most common source of primary infection. After that, management, field sanitation like removal of collateral host and infected debris from the field, use of slow release nitrogenous fertilizer is advisable. Then, grow tolerant varieties like O44 and Bhavani. Use disease free seeds. Then, treat the seed with Yaram or Captain at 4 gram per kg. Spray the nursery with Idifenfos 40 ml or Mancozeb 80 gram for 20% nursery, and spray the crop in the main field with Idifenfos 500 ml or Mancozeb 2 kg per hectare when grade reaches 3. If needed, then repeat after 15 days. Then the next disease is the thyroid disease. Basically, the thyroid disease, which goes due to the zinc deficiency. Dr. Y.L. Nene reported this disorder from UP, thyroidism, and yield loss up to 25%. First appearance of disease seen fortnight after transplanting, then yield up to 25% loss, then field appearance of disease seen fortnight after transplanting, and intervenial chlorosis. Numerous brown color lesions form cover cool leaf surface, and then ultimately leaves dried and died. Their management: 2 spray of zinc phosphate 2 kg plus slag lime 1 gram per kg. So 2 spray of zinc phosphate 2 kg plus slag lime 1 kg, which dissolve in 400 liter water, and spray first spray at appearance and second spray at 10 and second spray after 10 days interval. Now come to the diseases of cucurbits. The gourd family Cucurbitaceae consists of about 975 species across 98 genera. So the member of the family are the ground family Cucurbitaceae consists of um, consists of about 975. The members of the family are annual or perennial herbaceous plants. Then many are climbing or trailing winds with the characteristics of Tendrils, the flowers are often showy and unique, sexual and produce large fruit known as peppers. The important crop of this family are bottle gourd, watermelon, cucumber, muskmelon, squash, pumpkin. Then the important crop of this family are bottle gourd, watermelon, cucumber, muskmelon. So the diseases are like downy mildew disease, which caused by Pseudoparanospora cubensis. The host range like muskmelon, sponge gourd, and bitter gourd, yellow, angular spot restricted by veins resembling mosaic molting appear on upper surface of leaves. The corresponding lower surface of these spots shows a purplish downy growth in the moist weather. So the spots turn necrotic with age. The corresponding lower surface of these spots shows a purplish shown growth, downy growth in moist weather. The spore turn necrotic with age. So the diseased plants get stunned and die, and the fruits produced may not mature if and have a poor taste. So these are the symptoms of the disease. Now survival and spread. Primary oospores in soil and sporangia from perennial collateral weed host in the vicinity. Secondary wind-borne and rain-displaced conidia, sporangia or autonomous zoospores. Favorable conditions like relative humidity, that is 90%, high soil moisture, and frequent rains. So the destruction of cucurbitaceae weed around field, and spray of metal exile plus mancozeb 0.2% or copper oxychloride 0.3%. Now, next disease is a powdery mildew disease. The Causal organisms Aries field, Chichorinicidum, and Sphaerotheca fuligina. The host ranges like pumpkin, bottle gourd, coccinia, cucumber, and rich gourd. So the bitter gourd is less affected. The symptoms are like whitish or dirty gray, powdery growth on foliage, stems and young growing parts. The superficial growth ultimately covers the entire leaf area. The diseased areas turn brown and dry, leading to premature defoliation and death, and the fruits remain underdeveloped and are deformed. So, the fruits remain underdeveloped and the a- and are deformed, and the diseased are turned brown and dry, leading to premature defoliation and then death. So basically, these are the symptoms. Survival and spread. Primary survival, like dormant mycelium or clastothesia in infected plant debris or conidia from collateral host. Then the secondary is the wind-borne conidia. The favorable conditions are like the dormant mycelium or clastothesia in infected plant debris or conidia from collateral host. Then the secondary is the wind-borne conidia. The favorable conditions are like morning relative humidity, that is 90%, and the cool, cold, dry weather. These are the favorable conditions for the pathogen. Now, their management, like field sanitation, removal and destruction of collateral weed host. Then their management, like field sanitation, removal and destruction of collateral weed host from the vicinity of okay, so vicinity of the field. Then spray of tridmorph 0.1% or dinocap 0.2%. Next disease is the cucumber mosaic virus. So the wide host range, that is cucumber, pumpkin, the wide host range like cucumber, pumpkin, gourd, cowpea, tomato, and the cucumber virus with spherical particles having single-stranded RNA. Now, the cucumber virus with spherical particles having ssRNA. The symptoms appear on the youngest and still expanding leaves. When infection occurs, then 6 to 8 leaves. The symptoms are like symptom appear on the youngest and still expanding leaves when infection occurs at 6 to 8 leaves stage. Then the typical mosaic. So infection occurs at 6 to 8 leaves stage. Typical mosaic pattern which develops on leaves stage. The typical mosaic pattern develop on young leaves. Leaves curl downward and leaves curl downward and become motile, distorted, wrinkling, and the technician in charge. So when appear bunchy because of shortening of internodes, when infection occurs, then infection occurs at a mid-season. Previous growth remains normal and produce normal fruit. Then fruit set is very less. If infection occurs early in the crop growth and fruits are often mishapen, mortal way and reduce in size. The size of the leaf, which is short, and the leaves is wrinkled, and the color of the leaves is also changed. Survival and spread, like primary survival, virus particles on collateral and other weeds, ornamentals or crops, host banana flower, corn, passion fruit, cephalour, spinach, sugar beet, wild cucumber, Commelina cumenensis, Commelina diffusa, survival and spread. Primary survival, like virus particles on collateral and other weeds, ornamental crops, virus particle and collateral and other. So the host, banana, clover, and Shefla, spinach, sugar beet, wild cucumber, Commelina cumilis, then sugar beet, wild cucumber, then Nidiflora, solanum, Illiganifolium, Phytoplaza, The secondary is the virus particle. The secondary or virus particle transmitted by efforts and now peer management. Probe out the infected plants, then eliminate the perennial. Then management. Rogue out infected plants, eliminate perennial weed host, then raise the the rogue out the infected plants, eliminate perennial hybrid, and raise the barrier crops, vectors by vector control by spraying of monocrop. Then their management. Rogue out the infected plants, eliminate perennial weed host, raise barrier crops, vector control by spraying monoprotophos or phosphomethone 1.5 ml per liter of water. Then the Sarcospora leaf sport, the causal organisms Sarcospora citrulina, Sarcospora melones, and Sarcospora lignanium. The common on watermelon, muskmelon, and the common on watermelon, muskmelon, and cucumber. The symptoms are minute water-soaked spots or yellow specks develop initially on leaves. Spores enlarge rapidly and become circular to irregular with pale brown, tan, or white centers and purple to almost black margins. Spores colonies to form large blotches. The leaf may dry and die, presenting the leaf ice crust appearance, and fruits are also occasionally attacked. And these are the symptoms like on the leaf. Basically, the spores will be appear and they will be enlarged in the size and complete leaf which will be dry and the color of the leaf will be completely changed. These are the symptoms on the leaves. Then survival and spread. Primary dormant mycelium or conidia on infected plant debris or collateral host. Then secondary is the windborne conidia. Now their management. Maintain good soil drainage and good aeration between vines. Field sanitation. and to do 3 protective sprays with mancozeb 0.25% or carbendazim should be 0.1%. So this is all about the diseases of cucumber, sorry, diseases of cucurbits. Thank you.